Good afternoon. So my talk today is intended mostly for an audience who is not that familiar with cell sorting. So some of you may just not want to pay attention for the next few minutes. For cytometrists, I think a lot of people do flow analysis and don't necessarily do sorting. And for researchers who don't specialize in flow, I think there's a lot of people who come to the core labs to do cell sorting and just treat it as a black box. And I would like to have people improve their experiments by understanding a little bit better how the technology works. So with no further ado, I'm going to start my talk, hopefully. There we go. Okay, so the outline of my talk is a few slides about how sorters work and how they're different from analyzers. And then I'll have a lot of FAQ, which are questions that we get asked by every customer who comes into the lab and wants to sort cells. I want to stress how important sample quality is when you bring a sample to the core for cell sorting. And then we're going to look at some best practices and some of the cool things you can do with a cell sorter. So just a quick overview of the hardware for cell sorting. It's a pressurized system where you're essentially pulling or pushing sheath fluid or PBS from a pressurized tank down through a nozzle and flow cell. In the flow cell, you're injecting the sample, and you're using hydrodynamic focusing within the flow cell to push those cells into single file. And then there is a small aperture through which those cells pass. That is called the nozzle. And the nozzle is usually a small aperture in the range of 70 to 130, sometimes a little smaller, sometimes a little bit bigger microns. Then, as with any flow cytometer, you're going to have laser intercepts and detectors. I'm not going to talk too much about that. Mostly, I'm going to focus on the mechanics of the stream and drop creation, as well as how we're going to get those drops once they're charged into the collection tubes that everybody wants. Everything that doesn't get sorted comes out through a waste catch and goes into a waste tank. So it's pretty important to be focused on how the sorter works so that your cells of interest end up in your tubes and not in the waste tank. Before any talk about cell sorting, we should talk a little bit about the biosafety of cell sorters. Cell sorters are a special hazard for biosafety. There's an elevated risk because sorters can produce large visible clouds of aerosols that are sized pretty well to fit in, to go in for deep lung to go deep into your lung tissue. Anyway, so those particles will vary between about 50 and 500 nanometers. The 50 nanometer particles can stay in the air for a long period of time. So there needs to be protocols to assure that your aerosol containment has been validated. Your sorter should be placed in a biosafety cabinet in a room without additional equipment that should be used at the same time. You need to have negative air pressure, basically a tissue culture room. You should have respirators on hand where needed. And there should be project level risk assessment for cell sorting experiments. So in some ways, this part of flow cytometry is often best left to the experts. There are benchtop sorters, but do not neglect these risks or do so at your own risk. So there are two very basic types of cell sorters. And this is a historical sort of slide. The first is a jet and air sorter. These were developed in the late 70s. There are a couple of differences between a jet and air sorter and a cuvette-based sorter. A cuvette-based sorter uses a quartz cuvette and gives you a better signal resolution by passing your signal through the optically matched quartz cuvette and then generating the stream, whereas a jet and air sorter generates the stream and then actually reads out the fluorescence. That gives you a little bit more certainty in which drop is going to contain your cell of interest on a jet and air sorter, and a little less certainty on that on a cuvette-based sorter. So once you've gotten your fluorescent signal, you need to form drops. And there's a piezoelectric that chip that forms these drops at a resonant frequency. That resonant frequency is dependent on the nozzle size and the drop drive frequency. And then the amplitude is set to maintain a stable breakoff. That breakoff position, the last connected drop, needs to stay completely constant in order to actually accurately sort your cells. You'll see this stream camera on every cell sorter. The stream camera is viewed using a strobe and some image processing, which is why even though it's moving, it doesn't look like it's moving. If it does look like it's moving a lot, and you need to stop sorting. Critical for sort accuracy is to calculate the microsecond delay from the last sensing of the cell, so where the lasers intercept the stream, to where the last drop is connected. That value is called the drop delay. If your drop delay is incorrect or even a little bit incorrect, it can really reduce the purity and recovery of your resulting sample. And you can see this in a nice paper of which Rui Gardner is an author on a method called RMAX. Purity will stay pretty high across a reasonably large range of drop delay. However, recovery of your samples, how many sorted cells you get back out of the number you think you sorted, will drop off quite quickly. So your drop delay calculation and the accuracy of that calculation is pretty important in terms of getting back the cells you need out of the sorter. So next is droplet charging and deflection. Once we've identified a target cell, there's a charge that goes down the stream and breaks off into that first droplet. We can charge multiple droplets in order to increase the yield that we'll get off the sorter. Those charged droplets, once they have been charged, can be passed between charged plates. That deflects them left, right, and away from the uncharged droplets. Uncharged droplets will go to waste, as you can see here in the center, and you can get distinct side streams, up to about 6 of them on modern instruments on either side. Those are just a different charge level that's put into the stream when you go ahead and charge the drop. So the sorted cells should form a visible side stream on your actual sort. This particular one is just a test stream, but you should see your cells heading into the tube when you're sorting. So the metrics associated with cell sorting, we should be able to put numbers on all of these things. So everybody will think of sort purity, right? Sort purity is very important. That's the percentage of target cells that are in your sort product. So you should be able to approximate, in many cases, 100% purity. If you are not at 100% purity or close, you should be looking back and saying, how can I improve this experiment? The point of going through a cell sorting experiment really is to get your cells very pure for the next step of your work. Efficiency is the next thing that we like to think about with cell sorting. And efficiency is of the how many target cells were you actually able to sort out of the number that were present in your tube? Or what is the percentage of the sort count minus the abort count over the target events? I'll go through a few of those terms later on. But your efficiency is going to be part of the readout that you can see while you're sorting. It'll be within the sort stats. And where your efficiency should be will vary depending on the sample that you're running and the speed that you're running at. However, keeping that efficiency high is what's going to cause you to have good recovery. Recovery and yield is the next one. This is the percentage of sorted cells that are actually recovered post-sort. So if you took your sorted cells and you went and counted them, how many of those cells did you get back from the sorter? Lots of factors can actually cause problems with the yield and recovery count, because there's things you need to do to concentrate your cells and to count your cells that can have a lot of error in them. So I wouldn't be surprised if there's some differences between what the sorter says it sorted and what you actually were able to count when you got back to your lab. However, I think that those should be a predictable and consistent number, optimized to your method. The sort count is the other one. That's the absolute number of sorted cells, and that could be determined based on what the sorter gives you, or which is based on efficiency, or the yield or recovery, what you can actually see in your tube after you get it back. So now we're going to move to FAQ, and there's quite a lot of these because I've been doing this for a long time. So I get the same questions, and I've just put them into slides for all of you. Okay, so what are the buffers and media that we use in cell sorting? The first thing is sheath fluid. This is what we refer to all the time. Usually it's just buffered saline solution. On analyzers, you can use water, but on a cell sorter, because you need to charge your drops, you need to have conductivity. So usually it's just PBS or another type of saline. There's your sample buffer. What I would recommend to people is that they bring their samples in PBS with low protein. You can also bring them in media with low protein. You do need to make sure there's no clumps or particles of any kind. A lower protein than you use in complete media will help the cells, keep them from clumping and help them maintain viability, but it won't increase the viscosity, which can impact your sort. Your cell concentration will vary by application, but it usually varies between about 20 million and 500,000 per mil. You should, if you're using a core, consult with them, and if you're not using a core, see what advice you can get from experts to help you optimize these parameters and make the most of small samples or rare cells or any more challenging sort. There's also the collection buffer. This is the buffer that you put into your collection tube, and it's intended to cushion the landing because the cells come off the sorter moving pretty fast, and we don't want them to jump into an empty swimming pool. We want to have something in that collection buffer that's going to help them keep them happy. So often it's complete media. Some people will use high serum media. I personally used to sort into PBS plus 50% serum. This is a good way to go. You're going to dilute that. The more serum you can have in there, in my experience with lymphocytes anyway, they recover very nicely that way. You also can sort directly into an RNA DNA isolation buffer or an RNA later kind of thing. This will stop the cells at a point where maybe your transcriptomics are pretty stable and may be a good way to go depending on what you're looking to do. We have a lot of choices on that. The only negative on sort of an RNA DNA method there is that you're not going to be able to assay the cells after sorting. So you're just going to have to take on faith that you got back what you thought you did. And then the volumes for what you should collect into, at least 10 microliters in a 384, 25 in a 96, 200 microliters in Eppendorf tubes, 1 mil in 5 mil tubes, and 3 mils in 15 mil tubes. Now, we go lower than this all the time. You know, we sort into 4 microliters in a 700 microliter Eppendorf tube, but you really need to be very careful with targeting if you do that, and to make sure that your cells are landing in that collection buffer where they're going to be recoverable. So another great question, since everybody sees the drops, whether they know how the sorter works or not, the drops are up on the screen. Does every cell contain a drop? No, unfortunately, they don't. So there's some uncertainty in the cell position at the first drop break off that can influence the sort mode choice. For most sorting, about 1 in 4 drops can contain a cell, or they get too close. So if they get too close, they're not sorted, and that drops your efficiency, and you'll end up skipping those target cells. Those target cells that are skipped are called sort aborts, and they will be reported in any kind of sort report you can find. So the maximum event rate is going to depend on a lot of things. Basically how many drops are being made, which is the nozzle size, the pressure into the system, the drop drive frequency, and the amplitude. So on a 70 micron nozzle, you can get a max speed between about 16,000 and 20,000 events per second, depending on your drop drive frequency and your pressure. At 100 microns, you can get between about 6,000 and 8,000 events per second. You go higher than that, you're really just dumping more cells into the waste, and it's not, you end up kind of tipping point of you're using up your sample, but you're not getting more cells back. Okay, so a lot of cell sorters today use the term flow rate, and I find flow rate a little bit of a confusing term because the cell sorter has a speed that you can call a flow rate, but it's not a volumetric rate. So the cell sorter doesn't run 1 microliter per second or anything like that. It's a pressure differential between the sheath pressure and the sample pressure. So the sample always has to be injected into the sheath at a slightly higher pressure in order to push the samples into single file line in order to do hydrodynamic focusing. So as you increase that pressure differential, your events per second go up. That's why people do call it a flow rate. And the diameter of the hydrodynamically focused core stream also goes up. So as you can see on the right, that means your cells, as the core stream goes up, do not line up as nicely. So you lose some resolution in your fluorescence signals when your cells are not as nicely, clearly in single file. This can result in coincidences, particularly at high speed, as cells have more space to wiggle around in the core stream. So in general, I think you want to set up your samples to run at the lowest possible flow rate that you can while keeping them in suspension and not aggregating. And that is going to be an experiment-specific thing that you need to think about when you're planning. So another thing is, how do I set the threshold for cell sorting? I think most people in this room, and hopefully most people who are watching online, will know something about what a threshold is for flow cytometry. It's basically the minimum intensity value that you can set to cause the cytometer to think that something that breaks the laser beam is an event, not a cell, an event. Every particle below the threshold is just invisible to the sorter. This doesn't matter as much in some ways on an analyzer, but on the sorter, anything it can't see will be randomly distributed to the sort collection. And your downstream application will really have a lot of influence on what you want to, where you want to set your threshold. So it's often set on forward scatter height. You can also use fluorescence. That's application dependent. I've got two examples of a very low threshold here, where you can say that all my happy cells are right here. They're the nice purple ones. And I've got some tissue debris and other stuff, cell debris down here. In this case, I've got a higher threshold. Now, why would we do that in two different sorts? One reason that we would do that is because in this particular case, we're isolating cells for something where we want to be sure we don't get any dead cells into it. So we might be doing RNA-seq on these. In this case, we're going to do a co-culture experiment. And whatever's dead in that 7-day co-culture is not going to make any difference. You're not counting it when you decide how many cells you're putting into the wells. So those dying cells are just not going to make a big difference. That's, again, just application dependent. So think about what your downstream application will be, and that will help you determine where you want to set your threshold. For tissue debris and like digests of a tumor sample or something like that, you get so much tissue debris, you really have to be careful that you don't lose your cells within by setting the threshold. Okay, so another thing, some people, when I learned to do analytical flow, people weren't really doing as much doublet discrimination in analysis. I think in most cases, certainly people who work with cores at this point do doublet discrimination. This is a method where we use the raw pulse shape data of the event in order to determine whether we're looking at a single cell, which would have a higher height and equal width, or I'm sorry, and a higher, would have the same height and a increased width or area compared to the a doublet. And so the way that you set those gates is here you're looking at forward scatter height versus area after you've done your scatter gate, and you're setting a gate that is going to exclude these cells that have higher area than height, because those, because of the raw pulse shape, are going to be doublets. Now, doublets can be two cells stuck together, or if you're running fast, they can also be what we call coincidence, which is two cells that happen to be both counted as one event. For sorting, we usually use two sequential doublet discrimination gates. So you're also going to set gates a little bit differently in sorting versus analysis. In sorting, your gate logic is extremely important, and also the way you draw your gates is important. So for analysis gates, a lot of people use quadrants. It's not our favorite kind of gate in 2022, but a lot of people use quadrants, and this is a reasonable way to analyze this particular plot. If I were sorting these cells, I would want to be a lot more stringent in how I'm identifying these cell types. So for a sorting gate, if I use quadrants, I'm going to have a lot of cells that are on that boundary. We don't know what's going to happen with those. I'm going to have a lot of noise that's included. These are not extra big B cells here. This stuff here is not important to anything. We want to make sure that we're including exactly the cell types that we want. So we want to be pretty stringent and restrictive with the gates. Your gate logic is important because you can, more so than in an analysis method, run into a situation where you're trying to sort cells that are actually falling into more than one sort gate. So in this particular example of gate logic, this is from a BD Faxtiva instrument, and you can see that the sort populations for this were the naive cells for both CD4 and CD8, and these are mutually exclusive. However, if I tried to sort CD8 naive and CD8, I'm going to get a lot of cells that are not sorted because they belong to both of those populations. So you need to think through the logic and be sure that your gating is mutually exclusive, that none of your populations fall into branches of the same gating tree. Okay. Generally, I would set the gates so they don't touch because this will increase the aborts, and just make sure that you're very careful about the gate logic. Use double negative gates if you're moving to the next plot. So I'm going to give you a very simplified model of what a sort mode is. This probably was true years ago and is not exactly true now, but it's a good way to think about what a sort mode is. So a purity sort is the most common thing that people will think about. This used to be, and still is in some cases, a 2-drop envelope. What that means is that every time I find a target cell I want to sort, I'm going to sort 2 drops because I don't know if this cell, which is near the merge, is going to be in this drop or in this drop. So I'm going to sort both drops. I can also set up a four-way or multi-way sort. This could be a 1.5 drop sort. Now this case, if I were looking at this cell, which is centered in the drop, I might sort this one cell. However, if I was looking for this one, I would sort 2 cells because I had some uncertainty on whether this guy was going to break off down into this drop or come back up into this drop. Okay, so the sorter is not sorting half a drop. It's just making decisions about whether it's how many drops it's going to sort based on its certainty of where they're going to fall. There's also sorting mode that you'd use for plates or any kind of single cell sorting, which is often called single cell mode. This is a 1-drop sort envelope, and in many cases it uses a drop center or a cell centering portion to it, where it excludes cells like this one that are going to be not centered within the drops. So it's measuring where they are within tenths of the drops, and it's saying if it's in the first 2/10 or the last 2/10, we're not going to sort that. You'll miss a lot of cells if you do a single cell sort, but you'll also be very confident with what you get is what you expected to get. The last sort mode, which is kind of in many ways underutilized, although it's not for every sort by any means, is a yield or enriched sort. This is a type of sort where you can take a rare population and run it through and just cause there to be no aborts. So every target cell that you want to sort, regardless of what's near it, what's in the same drop, what's in the next drop, will be sorted. So coincidence events don't matter in that case. You just need to set your gate so that you're able to isolate the cells that way. And one positive thing about that is that it can be used for quick debulking. You can run a lot faster if you're doing it on enriched sort. So, and Vina will tell you a little bit more about sort modes in a few minutes. So that's all I have on that. So how can you expect 100% purity or near 100% purity when you're sorting a very low frequency population? Well, there is a drop in, you can get down to maybe 5% of your initial frequency per population and still expect high purity. But as you get below that, your purity is going to drop from 99% down to 95, 93. As you get to 1%, you're still going to be down somewhere in the 60s, 70s. As you go lower than that, in one round of sorting, you are going to start to have less purity. We can only enrich with the sorter. We can't purify if there are very, very, very few cells. But you can go from 1 in a million to 1 in 10. Okay, so you may want to think about enrichment strategies before sorting. And I know Simona Cola was telling you about max enrichment, which she does for her hematopoietic stem cells. There's also kits now that you can use for dead cell removal. You can do enriched sorting, as I mentioned. You can also increase your initial frequency by doing what's called index sort gating, and I'll show you an example of that in a little bit. So a few other fake graphs here. These are relationships between nozzle size, pressure, efficiency, and events per second. And what I'm showing you here is that as you increase your nozzle size, your sheath pressure always comes down. You need to find a resonant node, and your sheath pressure is always going to drop as your nozzle size increases. Your drops are bigger, and your pressure will be lower. Your efficiency is also dependent on your events per second. So I told you before that if you run near the maximum events per second at a 70 micron is about 20,000 events per second, your efficiency will drop from about 100% in many cases down to somewhere in the 70s. So you need to think about where you want to be in terms of efficiency. If you have plenty of cells to spare and you can afford to throw some away, you may want to run faster. It's all a balancing act. If you choose a different sort mode, this slope here is going to change. So nozzle size is also related to the maximum events per second, as I said before. So at a 70 micron nozzle, we can get up to 20,000 events per second. If you get all the way out to a 130 micron nozzle, you're going to be down in the 1,000 events per second maximum. So a bigger nozzle is great, and it's maybe less stressful for your cells, but it's also much slower, so your cells stay out of their native state for a longer period of time. It's a balance how many cells are going to come out alive if you really increase the nozzle size, drop the pressure, drop the rate, spend more time on the sorter. Okay, so that's all my FAQs. I'm going through quickly here. Next thing I'm going to tell you about is how to prepare a good quality sample. And this is not exactly a how-to, but it's some considerations on how you might approach this. So a high quality single cell suspension is the single most important factor that most people can control in their sort results. Possible things can happen with the sorter, but things mostly happen with the samples. If you have a sorter that's run by experts, you're much more likely to run into trouble with problems with sample preparation than you are to run into problems with the instrumentation itself. Okay, so don't clog the nozzle. This is the key to any successful sort experiment. This is the same diagram I showed you before, and last time I showed you this, all the cells were nicely lined up. Once they don't line up and something plugs up that 70 micron or 100 micron orifice, a clump of cells, what you end up with is a little bit of stuff can still pass through. So you get a spray of aerosol that comes all out of the sorter. It gets into the collection tubes randomly. It contains cells. It contains cell fragments. It contains whatever else happen to be in there that you do not want to be breathing. So clogging the sorter can easily ruin your experiment. You do need to really think hard about biosafety. If you're trying to be safe, just don't clog the sorter. There's simple things you can do to limit the chance that this is going to be happening. You're also going to contaminate your sort product in many cases. Lots of modern sorters have ways to close off the collection tubes if you do encounter a clog, but that still is going to impact the purity of the sort population. You're still not going to be able to close that off quickly enough with automatic sensing that you're still going to get a 99% pure population in your sort product if you've had a clog. Clearing the clog in itself is not just a hassle, but also can change some of the QC parameter factors with the instrument. So sometimes it doesn't come back to exactly the same place that it was. And that means that some of your parameters can change intensities. You may need to move gates a little bit. Your break off may move a little bit. Clog prevention, again, just please make your samples good enough that they don't clog the instrument. Start with a good quality sample, a clean sorter, filter your samples through 40 micron mesh before you load them on the sorter. And certainly look at them under a light as well. Make sure you don't see anything floating in the tubes. If you can see it, do not put it on that sorter. Okay, so your quality of sort samples is extremely critical for hydrodynamic focusing. It's critical for forming good drops because all kinds of extra stuff will make your drop shape change. It will cause them to behave strangely. They don't deflect as well. Contaminants in the cell suspension can include aggregated cells, free DNA, tissue debris, red cells, lots of things that can get in there. Your cells within the stream need to be staying centered, and any kind of larger bolus of stuff is going to stop that process. And that's going to impact your fluorescence signals as well as your actual sort. So some tips on how to get a good single cell suspension: filter everything last thing before sorting. Filter through 40 micron mesh every single sample before you put it on the sorter. Watch the stream break off. If you see it move, the sorter might be monitoring to look for it move, but if you can see it move, it's moving. Your event rate should stay stable during sort. So if you're started at 5,000 cells per second and it's bouncing down and up and down and up to 1,000, 7,000, take that sample off, vortex it, refilter it, put it back on, see if that helps. Consider diluting that sample. Use a low concentration of protein. About 2% FBS is good for a lot of cell types. If you don't like serum, there's some other ways to go with that. Also use 2 millimolar EDTA. It doesn't seem to hurt any cells that I have located, and it does inhibit calcium-dependent aggregation. And then figure out what you can do up front to remove your dead cells and tissue debris. And some of this may just be washing your samples repeatedly. If you can see pieces of debris, you're not getting extra cells out of that. You're just making your sort worse. Okay, so for sorting, you also should always be doing viability staining. You want to make sure you exclude dead cells from your sorts. If you are including dead cells, then you're always going to end up with sort of a progression where cells are dying, your numbers are inaccurate, and your dead cells are making the live cells a little less happy. Dead cells have unexpected fluorescence. Once they're dying, they start to fluoresce in weird places for reasons that are not related to your staining panel. You can use a couple of different types of viability stains. These would include DAPI, amine reactive live dead cell stains. These amine reactive dyes need to be stained with your antibodies. something like DAPI or Cytox or PI can be added at the sorter. If you don't forget that you need a single stain for your live dead control. It's a fluorochrome. It needs to have its own control. You will need dead cells in that control in order to do the compensation, and it can be a roadblock if you don't have any dead cells in your viability control. If you have very low viability or a lot of tissue debris, consider using calcium AM. Calcium AM is the only live dead marker that stains your live cells, not your dead cells. Always better to pick a positive signal than a negative signal, because most things that are events that are not cells are negative. Okay, so how do you adapt a staining method for cell sorting? You're going to want to stain, you're starting with a lot more cells in most cases. So you're going to want to stain in the smallest volume that you can get with your cells. So usually that works out to somewhere between 5 million for a big cell and 30 million per mil. Your antibody titers, if you get them from the companies, are usually weight or volume per 100 microliters of staining buffer, assuming 10 million per mil cells. So you're going to need to scale up the amount of antibody you use, but if you're using 1 million cells for analysis and you're going to use 10 million cells, you don't need to scale up by a factor of 10. You might want to scale up by a factor of 2 to get a decent titer. If you have an analysis method where you have very carefully titered your antibody, then you're going to want to scale up linearly. If you're using the minimal amount of antibody to get good resolution, then once you increase your cell number by a factor of 10, you're certainly going to need more antibody to maintain that good resolution. A lot of times people are starting for non-antibody methods with something that was developed and written, a protocol that was developed and written for analysis. And it's important to consider, again, that your cell numbers are probably higher for cell sorting, and you may need to think about things like time and volumes and the amount of dye available in order to get decent staining for this kind of approach. Okay, so we got through how to prepare a good quality sample, which is extremely important, and I hope everybody realizes the importance of this. This is the reason why experiments fail. Okay, so now we're going to talk about batch planning and applications of cell sorting. So I'm always going to tell everybody to plan ahead. Consult with the core if you're using a core. Look at the instrument that you're using if you're not using a core. Order your reagents. Make sure that the sorter you're going to use can see the fluorochromes that you've chosen. Write down a protocol. Outline the controls that you're going to need. Then run a pilot experiment, at least one. Try this before you run the real experiment. I have people who come into our lab all the time. They want to run something that's going to cost a lot of money, and they want to run it first time with all the mice they have. This is not a strategy for success. Try it first. Try it with something that doesn't matter. Identify the problems with the panels, the protocol, the antibody titers, and the controls. You're going to have enough trouble on the second experiment scaling up to a bigger experiment. So at least have run through with a successful result on the pilot experiment. In sorting, you often have to decide real quickly what you're going to sort. So in analysis, we can usually just record the data and then try to fix it up later. In a sort, you really need to kind of make a decision while your cells are still alive on what populations you're going to sort. So test your protocol before you start with critical samples. Often you can do pilots with things like PBMCs or normal mouse spleen, even though they don't exactly match what your experimental protocol is. They're normal cells that will stain with a lot of people's panels that are easy to get, okay? So we can help you find the cores, experts, anybody can help you find a way to test protocols without pulling up the very important cancer sample that you can't possibly replace. What kinds of controls do I need for cell sorting? Well, so the answer for this one, unlike almost every other question I've answered, is almost exactly the same controls that you would use for an analytical flow cytometry experiment, and all of them. So you need your unstained cells, you need beads that aren't stained if you're using single color beads, you need your single color controls. I like comp beads. I know there's a lot of provisos with them, but they're reliably, consistently is a positive population. And if you can't get a positive population, you can't do the math to do either compensation or unmix. Do not forget to include your viability stains. These are dyes. They don't stain regular comp beads. They don't stain live cells in many cases, so you have to have some dead cells. You need gating controls. These are usually FMOs. Once you've run your FMOs, you'll know where to place your gates more confidently, and you'll be able to proceed and with confidence that you're identifying the cells of interest. You should also consider all the biological controls that are relevant to your experiments. So you might have parental cells or wild type cells or normal PBMC or similar tissue that you might want to look at. If people are looking at mouse tumors, I always suggest that they look at mouse spleen because often they're looking at immune cells, and immune cells can be found in spleen, and spleen is easy to get and much cleaner than any tumor. So another fun question, how many cells and how much time do you need? I used to work with somebody who his answer anytime anybody asked him how many what's the minimum number of cells you can sort is 2. Got to bring 2 or you can't sort them. Anyway, so how many cells, the way to calculate this is relatively intuitive, but how many cells are you going to need for your downstream application? Take that, look at the initial frequency of your target cells. This should tell you how many total cells you need to bring to the sorter. Don't assume 100% yield, assume something like 70% yield. This just gives you an extra cushion to be able to say, I can still do the next step in my experiment. Once you know how many cells you need to bring to the sorter, you need to figure out what event rate is reasonable with your nozzle, your DDF, and your cells. This will help you estimate the time the sort will take and to optimize that cell per mil concentration to bring to this sorter. You can consider, we do this a lot in my lab, running in parallel on multiple sorters. Sabrina Bertolaccio told you yesterday about this, and she consistently will book 3 or 4 sorters for 3 or 4 hours all at the same time in order to get an experiment done. It's an efficient way to keep her cells alive and gives her good results and enough data and time that she's not spending 20 hours in the flow lab, and my staff is not quitting yet. Okay, so think about your specific downstream application. If you're putting your cells back into culture, which is like the old school, why would I sort cells? I'm going to sort them because they have GFP, right? If you're putting your cells back into culture, you can think about purity and recovery being possibly the most important things, purity probably being the most important. You'll need to find a compromise that's appropriate for your initial frequency. If you have very rare GFP positive cells that you're trying to sort, then you're not going to get to a 99.9% purity. So you should think about whether or not you can, what kind of compromise might be appropriate, whether you want to take those cells back and do another round of sorting after you've expanded them again. The cells per mil that you should bring for cell culture is going to depend on the nozzle size, which is somewhat dependent on the cell size for the cells you're going to sort. And you need to think hard in this case about sterility and carryover between samples. So make sure you clean the instrument with bleach and detergent, followed by water or PBS. It's always good to put it in a Dwyer on a tube of water. Make sure that you're not seeing events. You want to, if you're going to do single cell plate sorting, you have to have a pure population. You typically want 1 cell per well, so use your single cell mode. Bring your cells at a lower cell density, a lower cells per mil. And that would be so that you miss fewer cells because you're going to run slower. And again, consider index sorting if your cells are rare because that's going to increase your initial frequency. Okay, more modern things you can do, or more, I guess, fashionable things you can do with sorting. Plan for your specific assay. If you're doing functional assays, lots of cells do well after sorting for co-cultures, for all kinds of different functional assays. You may need to work it out for your particular cell type and your assay, but I would tell you that if anybody tells you that cells cannot be run in functional assays after sorting, I can prove them wrong. So consider sorting directly into the culture plates when it's appropriate, although do realize you'll be adding some PBS when you sort into culture plates. If you're sorting for animal models, either a cell line derived xenograft or a patient derived xenograft, you're going to need an accurate sort count, and you need that to be aseptic so that you're compliant with all the IACUC things. And if you're sorting donor cells from PDX or CDX animals that have already been produced, then plan your panel carefully and consider what you're going to do with those cells after the fact. It can be difficult to identify PDX and CDX cells, and lots of times in a patient-derived xenograft, those cells can be quite rare. So it's good to have a decent idea of what to expect before you get to the sorter. If you're purifying cells before omics, you're going to need very high purity and low collection volume. Your accurate counts are probably very important. So you might want to consider single cell mode for that, and that would include any of the omics applications that we discussed the other day, and I'm not going to cover that as well as they did. Okay, so applications for cell sorting. Now we mostly know how this works. So we can usually we often sort into tubes. These will be between 1 and 6 ways. I call this bulk sorting. So we're just not separating them as single cells. We're separating a bunch of types of cells into a tube. So for best results, bring your cells in a cell per mil, appropriate choice of nozzle. So I would tell people if they're sorting at 70 microns, they want to bring those cells to us between 20 and 30 million per mil. If you're sorting at 100 micron, you probably want to bring your cells at about 10 million per mil. It's always easier if you're in the flow lab to dilute your cells than it is to concentrate them. So bring some buffer to dilute them and make them a little bit more concentrated than you think you need them. Your choice of sort mode is going to depend on what you're doing with them downstream, how important is purity, yield, and single cell. And you can isolate up to 6 populations at the same time for a heterogeneous cell suspension. And we can also isolate as many as tens of millions of cells in a single sort. So we can sort as few as 1 cell. Sorry, we're coming back to that silly joke. And we can use about up to 40 floors to define complex phenotypes. The spectral sorting is coming online, and it's going to bring us up to that 40-ish space in terms of complexity, and we will keep you posted on that. So cell sorting is suitable for almost every kind of downstream application. So plate and slide sorting, we can do single cells or fixed number of cells. We typically will use a single cell sort mode for this. It just tightens up where the cells are going to land, as well as a few other things. It's an alternative to people do limiting dilution assays for CRISPR libraries these days, and clone isolations, and that leaves you culturing a ton of plates, and you may not want to do that. So putting them on the sorter and getting your cells of interest plated for you is a better way to get a single cell per well. For any kind of plate sorting, you're going to want your cells relatively at a lower cellularity. And I keep saying index sorting here, sorry. I'm going to go to that in the next slide. So you can use that for small samples like a patient biopsy. Some sorters can be set up to do simultaneous sorting of different populations into different rows on a plate. This is not especially common, but there are sorters that can do it. Check first to see if this is something that you need to do. Okay, so we can do a lot with complex phenotypes. This is actually data from Sabrina Bertolaccio's lab. And what we're doing here is we're looking at her myeloid cells, including a lineage cocktail, and we have 4 sort populations. So her non-conventional, intermediate, and conventional monocytes that come from the Lin negative gate here. So those are here in this plot. The Lin positive gate here goes to the MDACC. Both of those are preceded by CD66B negatives. So what we want to be sure here is that we have good gate logic. And the way I know we have good gate logic is that all of these gates are mutually exclusive. So these do not overlap, these do not overlap. And it's helpful if your lineage positive and negative are both drawn on the same plot, because you know they do not include the same cells in that case. So we want to make sure we're using mutually exclusive cells. In a lot of cases, you might want to use a gate that is a not gate in this as well. So she's got, starting with about 0.5% to 1% initial frequency, she'll get high enough purity to get downstream RNA-seq. And she uses a lot of controls, including biological controls, to determine the gate positions here. Vina's going to give a talk in a minute on rare cell sorting, so I was just going to say we can do rare cell sorting, and stay tuned for Vina's awesome talk. Index sorting, which I mentioned, this is kind of a cool thing. You can take rare cells that you would typically have trouble sorting, and you can set gates in such a way that you are using Boolean operators or a sort of hacked up Boolean operator. This is going to be an AND gate here that's shaped like an L. And you're going to say, all of these are my target population. Put them into a plate, it's single cell per well. And we're going to have an index where we can say, well A1 got a green one, and well A2 got a purple one. This is a good way to increase your initial frequency, which is going to increase your eventual purity of your sort product, as well as your recovery of your sort product. This is underutilized and is a thing that should be used in many applications. That retrospective knowledge of which cell went into which plate is also applicable to cells that are differentiating on a continuum, that are not exactly defined to a particular cell type. minimizes everything going to the waste if you are able to take this strategy. And we're hoping that soon we're going to be moving this into a multi-omics approach similar to what Tricia told us about yesterday. There are some roadblocks in multi-omics. I think we're taking a different direction than she had. We're going to be getting the Rhapsody system in the lab, and we'll be using that for multi-omics. Okay, and that's all I had to say, except to say thank you to the great people who work in my lab and the great people who used to work in my lab and all the amazing FlowTexans who are here today, as well as a few other people I worked with closely for cell sorting and learned a lot from. So thank you very much.
Okay, that was great. Just so you know, Karen, you had a lot of very positive feedback in the YouTube chat. And you've been volunteered to write a paper. So we're going to save, if there's any questions, you can ask them at the end because we're going to do a round table. And same thing, if there's any questions that are coming from YouTube, go ahead and put them in the chat and we'll get to them. Who's there? Okay. All right, our next speaker is Veena Papana. Veena is a flow cytometry coordinator at the University of Texas MD Anderson Cancer Center Advanced Cytometry and Sorting Facility. We've seen a few people from there. She received her master's degree in microbiology from the University of Bangalore, India in 2002. Veena has more than 10 years of biomedical and immunology research experience, including over 5 years of experience working in flow cytometry course, probably more at this point. She received her cytometry certification from ISAC in March 2015, and she's attended many hours of cytometry training from BD, ISAC, FlowCyte, Leica, Amnis, including a lot of software training too. She has been associated with the ACSF at MD Anderson since 2011, and today we're going to hear her talk about rare cell sorting, as Karen just mentioned. So please